Research and Graduate Education. And I'm here, I guess I could say, on behalf of the Council on Research and Graduate Education, which is an elected faculty council that selects all of the speakers at this lecture series. So John, you've truly been selected by a council of peers. It's a great pleasure to welcome you all back for this 2019-2020 series of Science and Medicine lectures. Chip Asbury is actually going to introduce John, and I'll ask Chip to come on up.
Thanks, John. So it's a real pleasure and an honor to introduce John. As most of you probably already know, John's an extraordinary scientist and a terrific person. And I just want to say a few words, so I'll keep it really brief, about why I think we're lucky to have John as a colleague. So I guess first thing is, John's CV is really quite impressive. He got his bachelor's degree in biology and anthropology from Swarthmore, and then he did his PhD working with Michael Reiser at Genelia Research Campus, studying how visual information is processed in the neural circuits of the fly. And then he did his postdoc with Rachel Wilson at Harvard, and there he made an unconventional choice to develop a completely new experimental preparation. Taking considerable risk, I would say, but that enabled him to develop a totally independent research direction that now forms the basis of the work that he's doing in his own lab with his team. Now, since founding his lab here at UW, he's won most of the early career awards that I've ever heard of, and some that I haven't heard of. Klingenstein, Simmons Fellowship, Sloan Fellowship, Searle Innovation Award from UW, McKnight Scholar, etc., Pew recently. So that's all impressive, but getting beyond his CV, what impresses me most about John is that underneath all his technical talents, he's unfailingly modest. He's really unassuming, and he's clearly driven by his own curiosity rather than any outside notions about what he's supposed to be doing. And in my view, this trait is what distinguishes great scientists. They retain this kind of childlike fascination with the world and around them, and they let that awe guide them in their actions. Now, John has a fascination with critters, especially insects, but also crustaceans and reptiles and bats and things. He recently told me about a new effort to try to study these tiny flies that you can find on snowfields in the Cascade Mountains. Maybe there's a fly in this picture here. This deep curiosity of John's comes through in all his research and when he talks about his work and when he writes about his work. And it's really infectious, and I've really enjoyed being part of that. Or hearing about that. John regularly writes commentaries about others' work. Often this is about work that's done on non-model organisms that aren't currently in the limelight of scientific research. And often it's about neural control of animal movement or perception. Now, I'm not a neuroscientist, and as an outsider, sometimes reading neuroscience papers feels like I'm accidentally wandering into someone else's party, like I'm at a wedding reception for people that I don't know. John's commentaries bring you right there into the party. He makes sure that you get a nice cocktail. He introduces you to all the guests. He even lets you in on the latest controversies and some of the gossip. So he's really masterful at encapsulating the essence of complicated topics in ways that are broadly accessible and also entertaining. And so I guess my ultimate point is that we're lucky to have such an excellent scientist who so skillfully shares his work, his understanding of his field, and his enthusiasm with all of us as one of our colleagues. So with that, I'll turn it over to John.
All right, thank you, Chip, for that very heartfelt introduction. It's a lot to live up to. All right. Pointer. Okay, so today I'm happy to tell you about what we've been up to in my lab for the past 3 years. So one of the motivating questions that drives our research is how it is that the body is sensed by the brain and how those signals are then used to control the body. And so I think that a really good litmus test for how well we understand something is if we compare the control systems from artificially engineered systems like robots compared to a control system that we might find in an animal like a human. So these are some kind of representative bloopers from the DARPA Robotics Challenge from just a few years ago. So the challenge here is that teams of engineers are asked to build a robot that will show up to kind of an obstacle course and complete the obstacle course. They know a little bit about what is going to happen, but they don't know the exact details. And what is obvious from these state-of-the-art robots, state-of-the-art a few years ago, and it's not much better these days, is that they're just utterly unable to deal with unexpected things in their environment. And so I think a good comparison to this challenge is when people show up to play a soccer game. They don't know exactly what's going to happen during the game, but they're able to flexibly adapt to the circumstances and move their bodies in a way that is virtuosic and quite elegant. And so I think a big question we can ask is, what is it about the control systems of animals that is so different from the way that people are designing robots? Now there are a lot of differences, but one that I want to highlight is how the architecture of animal control systems, so the nervous system that is controlling the body, is one way that it is very different from robots is that animals are extremely flexible. They have built-in feedback that they use to adapt on the fly. Whereas robots generally like to approach tasks by planning explicitly for the next movement that they're going to make. And so they need to know what they are trying to achieve, and then they calculate in a top-down way, like a computer calculating an outcome, what it is they're going to do, and then they execute it. And so if we look at the architecture of nervous systems, we see all these little feedback loops distributed throughout the body and throughout the nervous system that underlie flexibility. And so a kind of big scale answer to this question is that animals are using feedback from the environment to constantly adapt, whereas robots need to explicitly prepare for each movement that they're going to make. And so we can learn a lot from animals and how to design control systems like this in order to execute movements flexibly. The key sensory system that underlies our ability to move our bodies in a flexible manner is the sense of proprioception. So the sense of proprioception in our bodies is mediated by groups of mechanosensory neurons that are distributed throughout our muscles and our joints, so that when we move our limbs, for example, the contraction of the muscle generates forces that are detected by different populations of mechanosensory neurons that convert mechanical forces into electrical signals that are then transmitted down the axons of these neurons into the spinal cord. And so these trains of action potentials are encoding something about how it is that the body's moving, and then there are circuits in the spinal cord, and also circuits in the brainstem and in other parts of the brain. They use these feedback signals to then send signals to motor neurons to subsequently move the body. And this is a process that is happening continuously. In our bodies, we have different types of proprioceptive neurons, muscle spindles, Golgi tendon organs, and joint receptors that are detecting different aspects of a movement. And as I'll show you later in my talk, this is also a ubiquitous feature of other animals as well. Okay, so a central challenge to understanding how it is that these sensory motor circuits are controlling the body is the fact that it has historically been extremely difficult to record from the neurons that are performing these computations. So whether that be the proprioceptors or the inner neurons within the central nervous system that are actually transforming these signals, and the motor neurons that are then articulating the body. And this is because many of these circuits for limb motor control are embedded within the spinal cord, and it's just very difficult to physically access the spinal cord in any animal, let alone in a human. And so there's really a lack of understanding of the neural computations that are occurring in these kind of circuits. These circuits in vertebrates and in people are also extremely complex. We're talking about millions of neurons and thousands of cell types. And so it's really been challenging to take these kind of oversimplified schematics of how these circuits work and put identified neurons on them and understand what's happening at connections between specific neurons. So these are the motivating reasons for why it is that my lab studies a genetic model organism, the fruit fly, Drosophila. So the fruit fly is a tiny little animal that has amazing motor control. So the way that we often study this in the lab is that we take a fly and we glue it to a pin, and then we position it on top of a spherical treadmill so that we can track the movement of the treadmill. So here the fly is stationary, but the treadmill is moving, it's floating on a cushion of air, and then it's positioned in a virtual reality arena so that we can show visual stimuli to the animal and motivate it to move in different directions. And this allows us to record the animal's movements with high-speed video and then slow it down and track movements of the limbs to understand how it is that the animal is actually moving its body. So here we've slowed this video down by a factor of 20. And then down here I'm just plotting the angle of one particular joint, the femur tibia joint. And I show this here to illustrate a few things. One is that flies walk extremely fast. So each one of these cycles, the femur tibia joint flexing and extending lasts just 100 milliseconds. So they're taking about 10 to 20 steps a second. And another thing is that each step is slightly different than the last. So these are not just little robots that are executing the same movement over and over again. They might be turning, steering toward an object. They might trip over something and need to compensate. And so each cycle of this movement is slightly different and relies on feedback to plan the next movement. And flies use their legs for other things beyond just running. So I just wanted to show you kind of a buffet of interesting fly behaviors. So flies are able to navigate through the world using their limbs. And in this case, this fly is estimating the distance across a gap. It decides that it's far enough or it's short enough that it can reach across. It reaches over and swings itself over to the other side. And then there's also social behaviors. Flies have behaviors like courtship and aggression that all rely on the movement of the limbs and the circuits I'll be discussing. And then, as we all know, flies are very good at getting away from approaching predators like a fly swatter, and so flies can, within 20 or 30 milliseconds, detect an approaching object and launch themselves away from it. And then, of course, once they launch themselves away, they have this entirely different mode of transportation, which is flight, which again, they execute in an amazingly fast and virtuosic way. And so all of these behaviors, and many more, which I don't have time to mention here, are controlled by a nervous system that's about the size of a sesame seed. So the fruit fly's central brain, which is shown here, this is sustained against all the synapses, so we're just looking at the kind of neuropil of the brain. The central brain here contains about 120,000 individual neurons, and the ventral nerve cord, which is kind of analogous to the spinal cord, contains about 20 to 30,000 neurons. So even with this relatively miniaturized and obviously extremely well adapted nervous system, the fly is able to execute a wide range of behaviors. And the scale of this, 120 or 150,000 neurons, to me at least seems of the level of complexity that one could, for example, understand it within a human lifetime. Whereas something like the mouse brain or the human brain that contains billions of neurons, trillions of synapses, is a scale that is going to take much longer. And so the idea of one reason why we study the fly is that we take a reductionist approach, we have a system that can execute many behaviors we're very interested in, and maybe we can dive in deep and get mechanistic answers to questions about how those behaviors are controlled. And then of course the other reason we study the fly is that there's 100 years of genetic tools that have been developed for studying the fruit fly. So one tool that we rely on very heavily and I'll use throughout this talk is something called the GAL4 UAS system. And what this allows us to do is to label very specifically individual populations or cell types within the nervous system. So here, using fly genetics, we've labeled just a group of 5 cells that are in these recurring segments within the fly's ventral nerve cord. And we can label these neurons with a fluorescent protein. We can turn them on and off during different behaviors and try and understand their function. So these are the reasons why we study the fly. And the questions that we're interested in are how the circuits in the ventral nerve cord, like in the vertebrate spinal cord, are sensing proprioceptive signals from the leg, transforming them in the central nervous system, and then feeding them back onto the motor circuits that are actually controlling the body. So I generally break this down into kind of 3 research directions that we're pursuing in my lab, 2 of which I'll be discussing today. So first, we are trying to understand how limb position and movement is encoded by populations of proprioceptive neurons. And then secondly, we're starting to try and understand how the signals from the sensory periphery are integrated and transformed by circuits in the fly's central nervous system. And then lastly, we are trying to understand how these feedback signals from the proprioceptive system are used by motor circuits to allow the fly to plan and adapt for future movements. So today I'm going to start off by talking about some published work from the lab that just came out last year about neural coding of leg proprioception. And then in the second half of the talk, I'm going to be touching on how it is that the motor circuits are organized to execute behaviors like walking. Okay, so starting off with proprioception. So flies have, like vertebrates, have different types of proprioceptive neurons that are detecting different aspects of movement. These are the hair plates, the campaniform sensilla, and the cortical neurons. And they're each detecting different aspects of movement in a way that is somewhat analogous to what we find in vertebrates, in the sense that a dedicated neuron might detect position but not be sensitive to movement, or for example, the campaniform sensilla sense the load on the body but are not necessarily sensitive to the movement of a joint. The neurons I'm going to be focused on today are this population of neurons within the cortical organ. So when I started the lab, not much was known about these neurons other than they existed. And so we've spent quite a bit of time trying to understand how this population is encoding what the leg is doing. So just to orient you to where these proprioceptors are and what they look like, this is a group of 152 cells. They're located here in the femur, and then they're connected to the tibia. So this is the tibia, this is the femur. They're connected to the tibia by a series of tendons. So as the joint moves, it stretches the dendrites of these neurons. So these are the cell bodies labeled here in the femur, and so their dendrites are pulled this way, and they have mechanically gated ion channels that allow them to sense these movements. The axons of these sensory neurons then project into the fly's ventral nerve cord. So these are the same neurons as I showed you in the previous image, but now we're just looking at their axons projecting into the ventral nerve cord, and you can see they project widely across the nervous system. So one thing that we can do with the flies, we can ask what happens simply if we just manipulate these neurons in a walking animal. Now, this is a question that we just haven't had the tools to really investigate for quite some time. But now we have a suite of tools known as optogenetics that allow us to express a protein within a subset of neurons and then use light to take control of those neurons and either activate or silence them. So here we genetically express channel rhodopsin within these proprioceptors. We can then shine light on the leg, and in this case, activate those neurons. So here, this is just an example recording of optogenetically evoked activity in a central neuron in the fly. And then what we can do is ask, okay, if we put the fly on a spherical treadmill, like the one I showed you toward the beginning of my talk, and we activate or silence all of these neurons, what happens? Can the fly still walk without proprioception? And so the basic idea is the fly will be walking along, we'll have a laser pointing at the fly's leg, we'll turn the laser on very transiently for just 100 milliseconds or so, and just ask what the fly does, whether it's able to adapt to this stimulus. And this is a series of ongoing experiments in the lab that is much more complex than this, but I just want to show you one example to illustrate the importance of proprioception to motor control. So here we have a fly walking on the ball. This is the same fly, just from 3 different views, and we're filming at, I think, around 300 frames per second. So the fly will be walking along. When this little red dot comes on, that means that the laser, which is focused on the fly's left front leg, will turn on. And here we're expressing an option to turn on all the proprioceptive neurons for just 100 milliseconds or so. So the fly's walking along, happily, all of a sudden the light comes on, and you can see that the next step, it can't move. It's basically paralyzed. Its leg is fixed. The light turns off and it continues walking. So experiments of this type allow us to say, to just say something very basic, which is that that optogenetic manipulation of the fly's cortical organ, of these proprioceptors, messes with their walking behaviors. That proprioception is obviously critical to their ability to walk. This is something that has been tested in other ways in people, for example. There are humans that have had systemic infections that ablate all the proprioceptors from the neck down, and similarly, they're paralyzed. Even though their muscles are totally fine and their motor neurons are fine, they're unable to walk. And so this is just to illustrate really that these leg proprioceptors are really important for the fly's ability to move its legs and to execute walking behavior. Okay, but the main thrust of this project was that we wanted to understand what it is that these neurons are detecting about the fly's movements. So to do this, what we turned to was a technique called two-photon calcium imaging, where we express a genetically encoded calcium sensor in the axons of the leg proprioceptors and record their activity. And the way that this works is that there's a protein called GCAMP. It's a fluorescent protein whose fluorescence is proportional to the amount of calcium that is bound to the protein. So we can express this protein within a subset of neurons, in this case in the axons of the leg proprioceptors. And then we can scan a laser through the tissue with something called a two-photon microscope. looks something like this. So we scan the laser through the tissue, and then we collect the fluorescence that's coming back off of this indicator. And so that allows us to essentially optically record the activity of these neurons and read out their activity with the microscope. And it looks something like this. So here we're expressing this protein GCAMP in all the neurons in the fly's ventral nerve cord, and we're just scanning down through the ventral nerve cord. And you can see single neurons firing, turning on and off. Here the fly is just kind of waving its legs around. And this just illustrates what the kind of raw data from these experiments looks like. So to study proprioception, a central challenge has been that it's very difficult to deliver stimuli or to control the proprioceptive system in a way that you can understand what it is the neurons are encoding. And so this was the task that was given to Akira Mamiya, a postdoc in the lab, and he came up with this system that we now use all the time, where he glues a tiny little steel pin onto the fly's tibia and then moves the tibia around with a magnet mounted on a motor. So here's the fly in a holder under the 2-photon microscope. There's a little steel pin glued here. There's a magnet mounted on a high-speed motor. And so he can move the joint around so the femur is fixed relative to the tibia. So you can essentially control the angle of this joint. And then at the same time from below with high-speed video, we can track the angle of the joint. So you can start with the leg extended here to 180 degrees, flex it, and then bring it back again. It looks something like this. So this is actually on the 2 photons, kind of representative example of what the joint movements might look like. This is the movement of the magnet. You can actually see the laser scanning in the background, infrared laser. And here we're just moving the leg from extension to flexion, and then we'll go back in the other direction. So the reason that we're doing this is that we want to be able to ask, okay, when the leg is extended or when the leg is flexed or when we move in a particular way, how is it that the neurons are responding? What is it that they're encoding about the leg? And so this is an example of an experiment where Akira is doing these kind of leg movements, kind of ramping through different positions, while he's recording from the axons of all of these leg proprioceptors. So this little cartoon here shows the angle of the leg joint. So you can see at certain positions, for example, here when the leg is flexed, these pixels turn on. When the leg is extended, these other pixels turn on. And then there's another group of pixels here that are kind of flashing every time the leg moves. So if we take the activity within a region of interest like this, and we just calculate the changes in fluorescence, normalized by the baseline fluorescence, we get a signal that looks something like this, where you have certain positions, like when the leg is flexed here, where fluorescence is higher, or when the leg's extended here, and then you have these little flashes every time the leg is moving. So what Akira did was to use image segmentation methods to try and disentangle what these different types of signals are. So what he did was he took all of the pixels in the image, and he computed the correlations between those pixels, and then he clustered those correlations into groups of pixels that respond similarly to each other. And what you end up is a map that looks something like this. So these red pixels here all respond in a relatively similar way. They're active when the leg is flexed. These blue pixels here are active together, and they're generally active when the leg's extended. And then these green pixels here are the ones that are responding every time the leg is moving. So we have signals that are encoding the position of the leg, and then some that are sensitive to movement. So in these experiments, Akira is recording from a whole population of neurons. But what we can do in the fly is we can then dive in and identify genetic driver lines that label just the neurons that are encoding one of these features. And so that's what's shown here in this slide, are more specific driver lines that are labeling just subpopulations of neurons that are encoding, for example, in this case, these neurons we call the claw, these are the ones that are sensitive to position. So one subpopulation is active when the leg is flexed, another when it's extended. And then we identified a second group of cells that we call the club, and these are the movement sensors. They respond every time the leg moves, generating these kind of flashes to these stimuli. And then the third type we call the hook. These are similar to the club neurons in that they're responding to movement, but they respond only when you move the leg in one direction and not the other. And so they're directionally tuned to movement. So to summarize, what the entire population of leg proprioceptors is doing is that it's basically dividing up different types of proprioceptive signals, different types of leg movement into different channels. So we have position channels that are encoding whether the leg is flexed or extended, velocity channels that are encoding each movement, and then directionally tuned signals to tell you whether it's going in one direction or another direction. And so we think this is kind of the basic organization and the essential neural code for proprioception that is provided to the central nervous system and uses the feedback signal for controlling the body. So now I want to turn to that side of things, the motor control. So I've been telling you about the sensory side of things. These signals are then feeding back onto the motor system that are controlling the limbs. So we started at the other end of this schematic by studying the motor neurons that are actually controlling the leg. So the fly leg is composed of 5 segments, about 14 muscles, and then each of these muscles has many muscle fibers. And these muscle fibers are controlled by, or innervated by, the axons of motor neurons that control the muscles. So each muscle in the fly is innervated by 1 or more motor neurons. This is just an example of one motor neuron that's innervating the fly's femur. And importantly, it's good to keep in mind that the motor neurons that are innervating the femur, by contracting these muscles, they're controlling the tibia. So I'll be talking about motor neurons that are controlling the tibia, but just keep in mind that they are all innervating the femur. Okay, so this is what a motor neuron looks like in the central nervous system. This is the cell body of the motor neuron, this is the axon, and so that axon projects out here and then comes in this way. But when we started this project, there was very little known about the organization of the fly's motor system. And so in collaboration with Wei-Chung Lee's lab at Harvard, we've embarked on what has become a quite large project to use serial section electron microscopy to reconstruct all of the motor circuits that control the fly's legs. So the reason we're using electron microscopy is that with light microscopy, we can identify individual neurons, but we don't have the resolution with light microscopy to see the connections between them because they're smaller than the wavelength of light. So for that, we have to use electron microscopy. And what Wei's lab specializes in is the ability to take a piece of neural tissue like this and section it into thousands of sections, and then image each one sequentially. And then you might think that the fly's ventral nerve cord is this tiny, tiny little thing, but if you cut it into 1,000 sections and you image it, it takes about 3 months. You end up with 20 million images that are 162 terabytes. And then you have to stitch all these things back together because you've cut them into sections, and you have to align it all. And so this is what Wei's lab has done. And so we have a full EM volume of the fly's ventral nerve cord. So each one of these little tubes, this tangle of spaghetti, consists of the axons and dendrites of many, many different neurons. And then if we were to freeze it, you'd be able to see the synaptic connections between them. So the idea is that if you were to trace and reconstruct all the data here, you'd have what's called the connectome of the fly. You'd have the wiring diagram of all the connections between different neurons. That's obviously a big task. It's something that we're working toward. But to start off, what we did was to try and identify all the motor neurons that are controlling the leg. So what Jasper, a student in Wei's lab, did was to look at the leg nerve and to basically identify all the neurons going out to the leg. So it has a cell body in the ventral nerve cord and sends an axon out. It has to be a motor neuron. And then he led a team of about 15 undergraduates over the course of a year to trace all of these motor neurons. So these are all the motor neurons that are controlling the fly's legs. So you see that each leg has something between 60 and 70 motor neurons, a relatively tractable number, although slightly higher than we initially thought. And these are all the motor neurons that are innervating the fly's 2 front legs, which are the legs we primarily focus on for our physiology experiments. So from these populations of motor neurons, what we want to understand is how each of these individual cells is contributing to controlling the muscles and the movements of the fly. So a postdoc in the lab, Tony Azevedo, selected 3 of these motor neurons, all of which innervate the femur and control the tibia. They actually all are controlling flexion of the tibia. And so these are reconstructed biosynaptic fills of those 3 motor neurons. And then these are their axons out in the leg. So you can see that they're innervating different compartments and innervating different muscles within the leg. Tony developed a preparation where he could record from these motor neurons with electrophysiology as the animal was pushing on a force probe. So we can measure the force being produced by the leg as he's recording from these neurons. So he's recording from the cell bodies here in the ventral nerve cord, and then the tibia out here is free to move. And I'll show you a video of this in a little bit. And the first thing that he did was to characterize the basic intrinsic properties of these neurons. People hadn't recorded from leg motor neurons before in the fly, so we didn't know exactly what to expect. And what he found when he recorded from the fast, intermediate, and slow motor neurons was that there's a gradient of properties that spans these 3 different types. One of these properties is the resting potential of the cell. So we see here that the fast motor neuron rests more hyperpolarized than the slow motor neuron. We also see that the slow motor neurons have a high spontaneous firing rate, whereas the intermediate motor neurons rarely spike spontaneously, and then the fast motor neurons are almost never active except under very specific behavioral conditions. And then finally, the input resistance of the cell, which we calculate by injecting current into the cell and measuring the voltage deflection. We also find that there's a gradient where the fast motor neurons have much lower input resistance than the slow motor neurons. So this gradient, as I'll show you, exists across a number of different properties, and we think is kind of a fundamental organizational principle of how it is that these motor neurons are structured to control the leg. And it probably exists across many different joints. And another feature that Tony found a gradient in, and probably one of the most important ones, is force production. So what Tony did was that he would patch one of these motor neurons, so get an electrode on this tiny little cell body, and then evoke a single spike in the neuron, and then measure the force produced by the leg from a single spike in a single motor neuron. So here's the fly's femur, this is the tibia, this is the probe, and down here I'll be plotting the movement of the probe, and then the memory potential of the cell. And at the same time, Tony is doing a recording, extracellular recording from the same neuron out here in the leg. So when you do this, what you see is that a single spike in the motor neuron evokes a relatively large movement of the leg, something like 40 microns or so. This is actually enough force, which we can calculate from the calibration of the probe, to a force equivalent to the fly's complete body weight. So this is a single spike in a single motor neuron is producing a very substantial amount of force. If we compare this to the intermediate motor neuron, we see that a single spike in an intermediate motor neuron produces a measurable amount of force and a measurable movement of around 4 or 5 microns, but far less than the fast motor neuron. And then the slow motor neurons, these are the ones that have higher levels of spontaneous activity. You have to dramatically modify their firing rate to get changes in the fly's tibia movement. So here, Tony is injecting current to make the cell fire about 100 spikes. And you see that the movement that's evoked is about less than a micron. So if we summarize these force production experiments across a number of different cells and a number of different flies, what we find is that there's a gradient of force production where the fast motor neurons are producing a ton of force, the intermediate are intermediate, and then the slow motor neurons are producing very little force per spike. And the way we think about this is that these slow motor neurons might be used for more fine-scale dexterous motor control. So I like to think about it when the fly is like typing an email. These are the motor neurons that they're using. And then on this end of the spectrum, these are used to maybe generate more ballistic behaviors where they need to get away fast. And so they recruit their fast motor neurons in order to, let's say, take off or escape or sprint or something like that. And one prediction that comes out of this is that because these different motor neurons are recruited within these different force regimes, we hypothesize that there might be a recruitment order, that they're recruited in a sequential order, where the slow is generally active, and then if you need more force, then you recruit the intermediate, and then if you need more force, then you recruit the fast. So as a function of force production, you sequentially recruit these different neurons. And so to get at this question, Tony did some recordings from each of these motor neurons as the fly spontaneously moved its legs around. And he actually was able to do, in many cases, paired recordings, where he would do a wholesale recording from the intermediate neuron, and then get an extracellular recording in the leg from the fast neuron. And what this allowed us to do was to ask, okay, if we take a snapshot here where the fast motor neuron is just starting to spike, let's say it's fired one spike, we can ask what's happening in the intermediate and what's happening in the slow motor neuron. And so the expectation would be, if there is a recruitment order like this, that every time the fast motor neuron spikes, then the intermediate and the slow motor neurons should be active. And so that's what's shown here. This is an analysis to try and get that question. So Tony's asking, if we look at a single spike in the fast motor neuron, what is the firing rate in the intermediate neuron? So from this schematic here, you predict a single fast motor neuron, the intermediate neuron should be firing along at a pretty high rate, and that's what we see here. And if you look at a whole bunch of fast motor neuron spikes, and you look at the probability that the intermediate neuron has spiked in a short window before the fast motor neuron spike, we see that in 100% of cases, the intermediate neuron is always firing. We can then ask the same thing about the slow motor neuron. So again, when a fast motor neuron is spiking, the slow motor neuron should always be firing if there's a sequence. And again, that's what we see. On average, the slow motor neuron is firing around 100 hertz, and for 640 fast motor neuron spikes, we see that in all cases, the slow motor neuron has spiked in the previous 30 milliseconds. We can then also ask this question about the relationship between the intermediate and the slow. So if we do paired recordings from these two cells, do we see that a single intermediate spike, that when an intermediate neuron fires a single spike, that the slow motor neuron is always active? And again, we see that that's the case here. On average, they're firing at around 120 hertz. And if we look at all the spikes fired by an intermediate neuron, in 97% of the cases, the slow motor neuron has fired prior to the intermediate neuron. And interestingly, there is a small number of cases where that is not true. We think that's very interesting. But we think that for the most part, this recruitment order from slow to intermediate to fast holds within this population. Okay, the last thing I want to tell you about motor neurons is to kind of connect back to the beginning of the talk about proprioception. So these motor neurons are controlling joint movements that one might expect to be used during a behavior like walking, and so they obviously rely on feedback. And so Tony did some experiments where instead of allowing the fly to move its leg, he took control and moved the leg to evoke a response in proprioceptive neurons, and then recorded that response in the memory potential of the motor neurons. So here in the white trace, he's taken the fly's leg, it starts out at around 90 degrees, and he extends it, and in the gray trace, he flexes it, and then records a postsynaptic response, in this case in a fast motor neuron. And what we see is that indeed there are feedback responses in these motor neurons, but they're much smaller in the fast motor neuron than the ones we record in the intermediate neuron, and then the ones we record in the slow motor neuron. The slow motor neurons are receiving massive feedback signals that are basically continuously modulating their firing rate. Whereas the intermediate are getting some feedback, and the fast motor neurons are getting feedback of such an amplitude that it almost never influences their activity. So similar to the way that we see a gradient of intrinsic properties and force production properties, we see a gradient of feedback where the fast motor neurons are getting far less than the slow motor neurons. And the reason that we think this might be the case is that slow motor neurons are used to control various fine-scale movements like typing, right? So you need a lot of feedback to adjust your movements as you're typing on a keyboard. Whereas if you're executing a ballistic movement like jumping, there's less time and maybe it's less important to integrate feedback in that case. And then finally, Tony did some experiments to show that these signals at least partially arise from the cortisomal organ neurons that I talked about in the first part of the talk. So we think that the same signals we're recording that are sensing position and movement are contributing feedback signals to these motor neurons to alter their movement, alter their output, and control movement during different behaviors. Okay, so to summarize what I've shown you from our analysis of motor neurons, we find that there's a gradient of anatomical, physiological, and functional properties among motor neurons at this particular joint, and we think that this probably also extends to other joints in the fly's leg. And that the motor neurons within this gradient are recruiting in a sequence from the slow motor neurons that are producing little force to the fast motor neurons that are producing a lot of force. And the slow motor neurons receive a lot more feedback from the proprioceptive system than the fast motor neurons. So this organization, we think, has something to do with the flexibility and robustness of fly behavior. So dividing motor neurons up into ones that are controlling fast behaviors, ones that are controlling slow behaviors, and then giving them different amounts of feedback so you can control them in different ways, we think is probably a quite fundamental organization and probably has parallels across many different animals. Okay, so I thought I would conclude just by giving you a big picture of why it is that we're studying these questions in the fly and how the insights that we derive from these experiments might affect the world more broadly. So as Chip mentioned at the beginning, I happen to have a particular obsession with just understanding fly walking. I think that these tiny little animals that are able to execute these amazing and dynamic behaviors is just intrinsically fascinating. And so that's what motivates me to study this, but that's not what motivates many of the funding agencies, is my curiosity. So the idea for why we study these things in the fly is really the hope that taking a reductionist approach and trying to study a system where we can derive very deep insights, where we can very precisely manipulate things and understand the way that the nervous system works at the level of genetics and circuits, and linking that all the way to behavior, can identify general solutions to biological problems that exist across all animals. Many animals walk, and it's very likely that evolution has achieved similar solutions in the fly as in other animals. And really, because we can do things so quickly and so deeply in the fly, our work often provides a framework for investigation in other model species. And then finally, some of the control strategies that we identify by trying to understand the walking strategy of the fly have the potential to inspire artificial systems for designing robots. And then finally, there's also technologies that we're building along the way. I spend most of the time talking about biology, but we really spend maybe the majority of our time developing tools to be able to answer these questions. And so many of the genetic and molecular tools for targeting the nervous system have potential for translation and have been translated. And then also computational tools for understanding brain function and behavior. And so I want to just leave you with one future direction that is particularly exciting to me, which is a kind of extension of some of the experiments I showed you at the very beginning, where we're using optogenetics to manipulate the activity of proprioceptors in a walking fly. So just to remind you, the fly is tethered to a pin, it's walking on a ball. We use optogenetics to activate or silence particular neurons in the fly's leg. And what I showed you before was an analysis of what the fly's walking behavior was when we did that sort of manipulation. And basically I showed you the fly stops walking. But what we really want to be able to know is what's happening at the level of not just the whole animal, but really the level of individual joints and kinematics. So how is it that the animal's body movements are changing during these manipulations? And so we've just started to do this. This work is, these experiments have started to work within the past couple of weeks, actually. And so what we've been doing is developing tools in order to track in an automated manner all of the limbs of the fly as it's walking. So this is an example of one of those experiments where the fly is walking on a ball. And here we're using automated machine learning methods developed by Pierre to do 3D tracking of all the fly's leg joints in a way that requires no annotation by a human, except to originally train the neural network that does the tracking. This is a set of software tools called Anapose. It's built on a deep learning package called Deep LabCut. And basically the idea is that we track the legs from all these different angles, and then we stitch them together in 3D and use the three-dimensional nature of our data to correct for errors from the 2D tracking. And what we can then do is use these methods during optogenetic stimulation and ask how it's affecting the fly's behavior. We can also use these to try and understand how neural activity is related to the animal's behavior. So here we've started using Anapose to track the fly's legs while it's walking under the 2-photon microscopes while we're recording neural activity. And this is an example of an experiment from a postdoc lab, Chris Dahlman, who is imaging from a population of interneurons in the VNC as the fly is walking on the ball. And this particular population of interneurons, it turns out Chris discovered last week, actually turn off when the fly is walking and turn on when the fly is not walking, and we're interested in why it is that they do that. And these tools are not only useful for understanding fly behavior, but they are starting to have translational potential in other areas. So Pierre, the same student who developed the tracking software, has a company called Evolution Devices that is now applying these tracking tools to tracking humans for rehabilitation purposes. So the idea is that you track a human that is undergoing rehabilitation and use muscle stimulation to kind of correct their gait as they're walking. So this is just an example of how tools originally developed for studying tiny fruit fly legs might someday be used on you. I just want to thank all the people in my lab who did this work and point them out. So Tony's the one who did the motor neuron electrophysiology. Many of the fluorescent images generated here that I showed were generated by Pralakshya Gurung and Ann Sustar. Let's see, Akira did all the proprioception work. Chris is the one who did the calcium imaging I showed you in the last slide. Pierre is doing the tracking, and Evan back here is the one who has done all of our behavioral experiments. And I'd also like to thank our funding sources, our collaborators, and also my fellow members of the faculty here at UW who made it a really welcoming place and an excellent environment to do this kind of science. Thanks.
We have time for questions? Are there a few?
Yes.
Great talk. So you're looking at the connectome. Do you find that the neurons that control the legs are kind of copy-pasted to control the 3 different pairs of legs? Or is it all one center controlling the 3 different pairs of legs? And then how would you, depending on the answer, how would that scale to, vertebrates, right, which is only 1 pair of legs?
Yeah. So we don't have the complete answer to that question yet. What we do know, we've only traced the motor neurons at this point, and we think there are probably homologous neurons across different legs with for the most part, with some exceptions. But then there's all this other circuitry that connects intersegmentally between the legs. So we think that there is probably a lot of similarity across legs at the level of the actual, the motor neurons that are actuating the leg, but all the control circuitry we think is very unique. For example, there's not great evidence from anatomy, but from behavior, we know that flies tend to initiate walking with their front legs, and their front legs are kind of setting the pattern for the other legs. And so we think that things tend to flow from the front legs toward the rear legs. As to how this would work in vertebrates, I mean, similarly, there's segmental similarities between the upper limbs and the lower limbs in us or in a quadrupedal animal. There are similarities, but similarly, I think that the control architecture is probably very different for the different legs.
So would you hypothesize maybe that the back legs are getting a little bit of feedback from the front legs independent of the main brain? Like, within the brain,
Yes.
talk to each set of legs, so that the legs actually can talk to each other independent of the brain?
Well, we don't think the brain does very much for walking. So, I mean, you can get flies to walk without a brain. The basic walking pattern is generated within the ventral nerve cord. Obviously, the brain is important for things like turning and vision, all these other things. But we do think that feedback, I mean, I wouldn't say that we have good evidence for this, but my sense is that feedback generally flows from anterior to posterior. But I could be proven wrong. Yeah, sure.
Wonderful work. So I'm wondering also about the brain, and wondering whether, in addition to making the walking appropriate for what the brain is seeing, whether it could play a function in learning. To what extent is there evidence that the flies learn to modify their behavior on the basis of experience?
Well, there's a lot of evidence for them. I mean, flies learn in many of the same ways we do: visual learning, olfactory learning. Yeah, so they're learning in basically every sensory modality. But if you're talking about motor learning, so of the type that, for example, you've studied, like if you're trying to change an animal's motor trajectory, like a sequence of joint movements, we don't actually have great evidence for that yet, but I think it's just because we haven't studied it. So actually that's the next project that Tony's taking on right now, is what you can do in a primate or to some extent in a mouse is to train them to perform a given behavior and then perturb them. That's harder to do in a fly. But that's what we're trying to do is ask, can they learn to position their leg in a certain way relative to a punishment or a reward or something like that. But as far as the types of learning people have studied, I mean, you can get a fly to associate an odor with a punishment or a visual cue with a punishment or something like that. We think that's all happening up in the brain. Yeah, but there's probably, like in the spinal cord, there's probably plasticity at the level of interneurons connecting to motor neurons that is more low level and could underlie things like if you're walking along and you keep tripping, you eventually need to modify your gait to correct for that. That's the kind of learning we think could be happening in the ventral nerve cord and we're interested in studying. Yeah, Sharon.
So when you record the activity in the proprioceptor neurons, I was wondering if, sort of to follow up on this, if you could use the channel rhodopsin to perturb the cycle that you've recorded, sort of play it back along, and then see if the animal learns to correct its gait.
We would love to do that experiment. The issue with playing it back is just that there's about 200 different proprioceptors within a leg, and being able to, and they're probably all active during every given movement, and so that ensemble of activity is very difficult to recreate without the genetics. I think what we can do is these kind of giant gain of function, like turn them all on, turn them all off sort of experiments. That's what we can do right now. But the dream would be someday to have kind of genetic access and optical access to many different neurons and play them like the keys on a piano, like you described.
Are you able to systematically vary the visual input and make that offset from the reality of the movement of the ball and stuff like that. I'm just curious if you've looked at those cross-modal sensory connections.
It's not something we've studied, but people have studied it quite a bit. And so the simplest thing is, so the fly is walking through a virtual environment. You can change the gain of the fly's movements relative to the environment.
Right.
And flies will figure out that relationship across an extremely wide range of gains, almost immediately. And so people have studied that quite a bit. In the context of, it's mainly been studied in the context of flight. But we have not, I would say, we have not really looked at the integration of where proprioception intersects with that. But obviously they would need to. You need to know how you're moving your body relative to how the world is moving around you.
Right, and that was part of my question is to what degree, I study children, so it's a different world for me, but volition and the animal's sort of intended action versus what they expect versus what the feedback you're getting proprioceptively and how that aligns and
Yeah, so within the past 3 or 4 years, there's been an amazing amount of progress on the spatial navigation circuitry in the fly brain. So we now know exactly which neurons represent which way the animal's trying to go. And you can use these methods to record from those neurons and basically figure out which way the fly is trying to walk. And so you can use that, I mean, this gets kind of into the weeds, but you can record those signals and then like mess with their proprioceptive feedback to try and understand how it is they're compensating and then how that actually translates into behavior. And there's a lot of very beautiful work that's been done recently on that in the fly.
one last part, is there any developmental sort of lens to this as well in terms of what experience needs to happen for these to function properly?
Yeah.
Versus...
So flies, as far as we know, they emerge from metamorphosis completely able to walk. There's probably fine-tuning of the walking pattern. I mean, that's true of many animals, that they can walk right off the bat. The thing we do know is that during development, there's patterned spontaneous activity within the nervous system that probably structures these circuits to allow them to work initially. But we actually have not spent a lot of time looking at how experience is changing, for example, walking gait. But that's something Brandon, a grad student in the lab, he's just started to look at those sort of questions. Yeah. It's brutal.
With respect to the first question, and your answer that the forelimb is sort of instructing the middle limb is instructing the rear limb, I was reminded of a study that was possibly in Nature 5 or 10 years ago of how quadrupeds, like mules or something, walk on very narrow ledges and keep going. And so it turned out that by analysis they found that the forelimb went down in some place that presumably the eyes had chosen as good. And that the rear limb ended up in exactly the same spot in the walking, so that it didn't have to be analyzed.
Yeah, interesting.
I don't know whether that could all be done in the spinal cord or whether it requires the brain, but I thought it was really helpful.
Yeah, no, that's amazing. Yeah, we have not put flies on treacherous cliff scenarios, but what we could do is just analyze, and we have the data to do this now. Just analyze their footfalls and ask basically, is the rear leg always basically falling into the spot where the front leg kind of initially placed itself? Yeah.
I think you as a mountaineer would appreciate this.
Yeah. Yeah.
Any other questions? If not, one piece of business, John, a little souvenir.
Ooh, a certificate.
You put it in your promotion packet or hang it and stand up.
All right.
One other thing too. Members of the lab group here, okay?
They better be here.
I want you to know that as a new investigator award in this series, that the investigator receives a little kick in a research account of $2,500, and you guys should determine how that money's actually. It can't buy beer, okay, but it can buy coffee machines.
All right. Thank you.